Welcome to the Human Ideasphere series, Chapter 9, Life, Part 1. We last left our Earth covered in water with mild temperatures. The oceans became the cradle of life, providing an environment where a variety of inert materials could interact and evolve. 4 types of matter emerged as crucial to the development of life. Lipids are molecules that repel water on one end and attract water on the other. When these molecules are strung together, they form a protective barrier that keeps water away from the inside. A single layer, called a monolayer, mixes with the water inside. A double layer, or bilayer, will repel water once it gets inside and keep the lipid layer dry. Carbohydrates are molecules made up of hydrogen, oxygen, and carbon. Carbohydrates are easily broken down into simple sugars. Glucose is a very simple sugar that can be converted into energy. Amino acids are molecules made up of hydrogen, oxygen, carbon, and nitrogen. They can form long strings we call proteins. Depending on their shape and structure, proteins can make all kinds of things like bones and blood and tissue. Collagen is an essential protein made from 19 types of amino acids. Nucleic acids are complex, nitrogen-based molecules hooked together in long strings. They are made up of nucleotides, which have a phosphate group, a sugar group, and a nitrogen group. The 2 most famous nucleic acids are ribonucleic acid and deoxyribonucleic acid, or as we commonly call them, RNA and DNA. These molecules, swimming in the primordial oceans, began to interact in countless combinations. Most of these combinations were unstable and short-lived, but some were able to remain in their joined state. Stop for a minute and take a look at how the early oceans looked. Does this remind you of something? Doesn't this look suspiciously like our early universe? Numerous stars and galaxies dispersed in a vast universe. Numerous particles dispersed in a vast ocean. This is how change works. Small bits thrown together coalesce to make bigger bits. The material that gets affected does change, but the pattern never changes. About 750 million years into Earth's existence, a particular combination of materials emerged that had staying power. This object had a round shape due to an outer shell made of lipids. The lipid shell repelled water on the outside, but contained water on the inside. Proteins could open the shell temporarily to allow water and other molecules to enter inside. Inside these early structures, RNA played a crucial role. RNA is highly reactive. Do you see the nitrogen base here? This end of the RNA molecule could react with all kinds of things in the enclosed space of the lipid. An important thing RNA could do was react with amino acids to create proteins. RNA could also copy itself, though the process was indirect. Let's go through this because it is crucial in the formation of life. The nitrogen end of RNA is very reactive, as we have mentioned. But these nitrogen bases also had a serious limitation. Each base could only bond with one particular molecule, called its complement. This means that an RNA strand would always bond exactly the same way each time it underwent a reaction. Because the bonds holding the RNA bases are weak, they unravel easily, leaving 2 strands, an original and a complement. When the 2 strands react and split again, they make 4 new RNA strands, 1 original, 2 complements, and another copy of the original. As you can see, it takes a few steps, but RNA was able to make an exact duplicate of itself. Because RNA and DNA are so structurally similar, RNA could also react with DNA and cause proteins to be made from the DNA strand. Occasionally, certain types of RNA molecules could react with a DNA strand and cause the entire DNA strand to copy itself. This ability of RNA to copy itself and DNA was a game changer.